to turn a gene into a protein. We've taken a look at how cells can turn on and off genes. We've taken a look at what kinds of mutations can occur. We've taken a look at how cells can fix mutations. So what we'll take a look at here is we'll kind of tack on one more tidbit which you've looked at so far. And the theme of what we're going to be talking about for the next 15 or so minutes is just cool things that bacteria can do with their DNA. That's going to be the theme. Cool things that bacteria can do with their DNA. You know what this is right here, Shane?
It's like a bacillus. Looks like E. coli.
I was thinking more green Cheeto.
Oh.
Right. So if you eat those, then your fingers turn. Oh, also looks like a condensed chromosome. It does. But so those are actually Pseudomonas. It's a bacillus. And it turns out that, so the reason I have that up there is because these bacteria have the capability of turning on and off genes depending on whether they have a lot of other bacteria in the environment with them. So it turns out that that's one of the cool things that we're going to take a look at next. And yeah, I'm going to do it right now. So we need to frame this, to set this up, what we're going to take a look at next, we'll kind of review really quickly one of the things we took a look at before. And before we took a look at an example of how bacteria can turn on and off genes, we looked at the lactose operon as the example, it's the prime example of how genes in bacteria can be turned on and off. And we said that basically what turns on the lactose operon is if there is what in the environment? Lactose. Yeah, so the presence of lactose causes these genes to get turned on, basically because some things happen that cause that repressor protein to fall off, which then allows the RNA polymerase to transcribe all of those genes. So that's one way that bacteria have of turning on and off their genes, but it turns out that that's not the only way that bacteria can turn on and off their genes. So what we'll show you next is just another method for turning on and off genes in bacteria, and it's a method called the two-component regulatory system. It's called that because it has two parts to it. Does that make sense? Two-component because it has two parts. All right. Here are the two parts. If that represents a bacterial cell, here are the two parts. One of the parts of this regulatory system is found within the membrane, and then the other part is found in the cytoplasm of the cell. The part in the membrane is called the sensor, and then the part in the cytoplasm, the pink thing, is called the response regulator. What the sensor does, you might guess, is that it can bind to, it can sense signaling molecules in the environment of the bacterial cell. And so examples of things that can serve as signaling molecules are nutrients, or maybe other molecules that are released by other bacteria in the environment. But it responds to some sort of signal that it's specific for. And after it binds to it, it causes that sensor to change shape in a way that allows kind of a docking spot to open up within that sensor on the inside of the cell. Once that docking spot has opened up, what ends up actually docking there is a molecule of, you know what that is? A molecule of ATP, and that ATP dumps off one of its phosphate groups basically to the sensor, which then gets transferred over to the response regulator, like this. And it turns out that once that response regulator has a phosphate group to it, or attached to it, it's considered to be in its active form. And what it can do when it's in its active form is to attach to a piece of DNA, just upstream of a gene, and depending on what kind of response regulator that is, that can either turn that gene on or turn that gene off. But it regulates what that gene does. So this is just another system that bacteria have for turning on and off genes. I'm going to sum it up. Here's what we said. First, the sensor that you find in the membrane binds to a signal that's in the environment of the cell. That then causes a phosphate group to get attached to the sensor. And so the fancy word for adding a phosphate group to something is that it gets phosphorylated. That phosphate group then ends up getting transferred to the response regulator, the pink thing in the picture. then once that has a phosphate group to it, or on it, it can then attach to the DNA, which then causes a gene to get turned on or off.
Where does it attach?
Just upstream of a gene. So we're not gonna, I don't think there's a specific name to the region where it attaches to, because it depends on the kind of response regulator it is, but it's just upstream of the gene. Alright, so any other questions on this? I mentioned that an example of a signal that can cause all of this to happen is a molecule that is released by other bacteria. So what this means is that because there's a system like this in place, it's possible for bacterial cells to communicate with one another and to actually influence whether or not they turn on a certain set of genes. So here's the reason why that is. Right, so say that there's just two bacterial cells within this environment, which is the screen. If they're going to try to talk to one another by releasing these signaling molecules, if there aren't a lot of them around, like you see up on the screen, chances are not too good that that will find its way over to that bacterial cell sensor, at least not anytime soon. But on the other hand, if there's just a lot of bacteria, if bacteria are really dense within a certain environment, and if they're all releasing these signaling molecules, then in that instance, just because there's a lot more signaling molecules, chances are better that one of them will end up on the sensor of another bacterial cell. So I guess what I'm trying to say here is that and whenever that happens, whenever a bacterial cell receives one of these signals, it causes it to change its behavior by turning on a gene. So I guess what I'm trying to say here is that there are certain things that bacteria do only if there's lots of other bacterial cells around. So the likelihood of one cell receiving a signal from another is greater if there's a lot of cells in the environment, just because more bacterial cells means more signaling molecules that are being released. And the official term for this, whenever bacteria do something, but only when there's a lot of other bacteria around, it's called quorum sensing. So again, quorum sensing is a change in cell behavior that happens only when there are a lot of other cells nearby. Basically, it's cellular peer pressure. You do something only when you know that all these other cells are around, kind of doing the same thing. And the reason why this is kind of a useful thing for bacteria is because it helps to save energy in some circumstances. And that's because there are certain things that are only worth doing only if you have a lot of friends around. For example,
Streaking.
that's only worth doing if you have a lot of other friends around. Maybe that's where me and you differ.
Fair.
Yeah. So transformation, that's a good example. That's a good example.
Yeah, because that'll make them
Yeah.
competent, right? Or quorum sensing can help aid in their competence.
Yeah, transformation makes sense. So what is transformation again?
When a free-floating DNA fragment gets inserted into a bacteria cell, only one strand of it, and then it's able to replicate it, and then since it's only one strand, then only one daughter cell will have that new gene.
Right. Yeah, nice. So the ability to take in naked DNA, that would be something that would only be worth doing if you know that there's a lot of other cells around, because that's the situation where there's probably a lot of DNA to take in. And so it'd be kind of dumb if you were just a cell all by yourself to turn on the ability to take in DNA if there's no other DNA around.
Or activating a mutagen, like promoting mutation.
And why would that be useful if to do only if there's a lot of other bacteria around?
Well, if there's a whole bunch of things around, then it would be worthwhile to risk a change to a fraction of the colony that may either eliminate it or cause it to survive an epidemic that destroys all the rest.
Right. Nice. I don't know of any specific examples of that, but that makes sense. Yeah. Cool. You guys came up with some good examples. I was going to go with the wave. Has anyone ever tried doing a one-person wave? Yeah, Austin.
Every morning.
When you get out of bed?
Yeah.
Does it do you pretty good?
It gets me going, but
It does?
it's hard to do with only one person.
You're like messing up my whole hypothesis.
It's not
You're
my
supposed
intention.
to say every morning when I do it, I feel really dumb afterwards.
I do.
Okay, so that is a perfect example of what we're trying to illustrate here. Like if you do the wave all by yourself, you feel kind of dumb. But if you have a lot of other people to do it with, then it might be worth doing, because then you get something cool happening, kind of like this. Yeah.
Nice.
Isn't that a lot cooler than just doing it by yourself?
Yeah.
Yeah. So just an example of when it might be a waste of energy to do something by yourself, but if you do the same thing with other folks, it's not a waste of energy anymore because you actually get something cool out of it. You guys mentioned some examples of bacteria. I thought I'd show you just one more because it has pictures to it. It's something that has to do with this guy right here. It's something called a Hawaiian bobtail squid. And you might notice right off the bat that what makes this kind of unique, and one of the things that makes this animal cool, is that it lights up. And the reason why it lights up is basically it helps it to get food. So you spare no expense when it comes to the factors in this course right here. So say that this is a Hawaiian bobtail squid, and it's in the middle of the day, and he's hungry. What's that?
It's majestic.
Thanks. Yeah, and yeah, you might be surprised how long it takes a brother to put something like that together. Even though it looks kind of kindergarten. So say that that's a Hawaiian bobtail squid. It's in the middle of the day, and he goes, "Oh, I'm kind of hungry. Maybe it's because it's lunchtime." If his goal is to hunt for, say, a smaller fish and to eat it, it might be tough to do because if it's in the middle of the day, the jellyfish or the squid ends up casting a shadow, which means that if he goes hunting for prey, the prey kind of knows that he's coming because all of a sudden it gets dark, and so he swims away.
Wow.
Why does the fish look red?
I think I could teach zoology too. All right. So the reason why it's an advantage, a good thing, for this guy to light up is because if he's lit up during the day, then he doesn't cast a shadow, which kind of makes him more stealth during the day, so that he can catch some fish to have for lunch. So it's a useful thing. So the reason why they make, if you hunt it only at night, you might be able to get around this this dilemma, but I don't know why they don't just do that. Maybe they don't have big enough stomachs to just store food at night and then have them go for the rest of the day. But that's the reason why it's useful. At least that's the reason why it's hypothesized that it's useful. Here's how it relates to bacteria and quorum sensing. The reason why these squid,
Does it also hunt at night? Or does it have a big enough stomach to only eat during the day?
I would imagine that it also hunts at night. Yeah,
But
hunts
does
at
it-
night, so it has to have a way of turning that off at
Okay.
night, because then, yeah, it just be this big, you don't come in, I'm lit up. Yeah, it'd be pretty conspicuous.
Yeah, so it's okay. You can turn on.
Yeah. So the reason why these guys can light up is because they have an organ that lights up. And the reason why that organ can light up is because it contains a bunch of bacteria that individually can light up. It turns out that those bacteria only spend the energy to light up if there's a lot of other bacteria in its environment that can also light up. So the idea is that if you're just a single bacterial cell and you're going to spend all this energy to light up, that's still not enough light to cause this squid to be able to hunt and get food. But if there's a lot of you kind of together collectively producing light, then that's enough light for this to capture food, which means that the bacteria living inside of it also gets food. So just one example of quorum sensing, it's a type of bacteria called Vibrio fischeri. So Vibrio fischeri perform bioluminescence, just means that they light up, but only when there's a lot of other bacteria around. That's an example of quorum sensing, which is one cool thing that bacteria can do with your DNA. They can turn on and off genes only if there's a lot of other bacteria around. You ever wonder what a Hawaiian bobtail squid tastes like?
Delicious.
Yeah. And if your poop would light up after you had something,
That's a good question.
probably at least temporarily.
Did you hear about that plant that they engineered so that it glows? It's like a fern, and you can buy them.
I have. I don't know which gene they use though to make it light up. Do you?
No.
Maybe a gene from Vibrio for sure. All right. Any other questions on this? Have you ever had one of these? All right. Here we go. So that's just one cool thing that bacteria can do with their DNA. Here's cool thing number 2 that bacteria can do with their DNA. Not necessarily related, except for the fact that it's just another cool thing. Turns out that this actually not only happens in bacteria, but you find this in every cell that has DNA inside of it. It turns out that bacteria can make certain, or they have certain genes that can switch places within the same DNA molecule. So that's what this is doing. That's what this is showing here, is that there are some instances where there's a gene in the DNA that can go, oh, I don't like being in this place anymore. Let me move to a slightly different place in the DNA. And there's a term to describe these genes that can move from one spot in the DNA to another. Those genes are called transposons. Again, a transposon is a gene that can move from one part of the DNA to another part of the DNA. And to kind of tie this into what we looked at during the warm-up, in the warm-up we saw what kinds of things can cause mutations. Right? We saw that things in the environment, like chemicals and radiation, can make it more likely for a mutation to happen. Is it likely, or is it possible for a mutation to happen even if there are no chemicals or radiation? It is, yeah, so just random mistakes by DNA polymerase is another thing that can result in a mutation. It's those spontaneous mutations, just random mutations. And so what I'm saying here is that this is another thing that can make it more likely for a mutation to happen, these transposons. And so what we'll take a look at next is kind of how a transposon can result in a mutation and some examples of things that transposons can result in. So first, how is it possible that a transposon might cause a mutation? So again, what transposons are by definition, it's a gene that can move from one part of the DNA to another part of the DNA. So here's a bacterial cell. Here's a chunk of its DNA. That red thing represents a transposon, a gene that can move. And so by definition, if it's a transposon, here's what it does. It can leave the part of the DNA that it's in and insert itself into a different part of the DNA. So what I'm saying is that that can result in a mutation because sometimes transposons jump right into the middle of another gene. So when that happens, it disrupts the sequence of that gene, causing it to, in a lot of cases, not be able to be read. Eric?
Are transposons actual functional proteins all the time, or are they just random codons? That's just a segment.
That's a good question. So in a lot of cases, they're just random bits of things. They're defined as genes that can move from one place of the DNA to another, but a lot of times they don't have their own promoter, so it's hard for, or it's not possible for them to be turned into a protein themselves. Yeah. So
is that their only function, to introduce randomness to the genetic code?
So there's some discussion as far as the reason why transposons still persist in our cells and living things, because it seems like it's doing something kind of harmful. So if it's doing something harmful, you'd think that it would be kind of naturally selected out. But you still find transposons in a lot of, or in basically all cells. So one of the thoughts out there is, just like you said, it's actually a good thing to introduce variation, like transposons can do. So for example, we'll see later that we have the ability to produce many thousands, millions of different types of antibodies to protect us against whatever virus or bacteria we encounter. And there's a thought out there that there's something like a transposon that might be involved in allowing us to produce so many different types of antibodies. So it looks like a bad thing, but there could be some useful things that come out of it. So let's say this. So transposons, they can jump from one part of the DNA to another. And when they do that, they can cause other genes to get messed up. Like particularly if they jump right into the middle of another gene sequence. And it turns out that there's some examples of phenomenon that are caused by having a transposon jump right into the middle of another gene. One example, the first example will show you something that is seen in people. It turns out that it's a type of hemophilia that's caused by a transposon jumping right into the middle of another gene. It's a type of hemophilia called hemophilia B. And basically, with hemophilia, you are not able to produce a certain type of clotting factor, which makes it tougher to stop any bleeding if it happens. And it turns out that with hemophilia B, the clotting factor that is involved, the one that's not produced, is something called factor 9. Right, so in factor 9, what this gene normally does is that it makes a clotting factor called factor 9. But in those with hemophilia B, they're not able to produce that clotting factor. And the reason why is because if you take a look at their gene for that blood clotting factor, there's actually a transposon that has jumped right into the middle of it, which keeps it from getting read properly.
Would that be one of those things that we could CRISPR out?
Possibly, yeah. I mean, that seems like a prime candidate for CRISPR if it's just caused by,
Just
yeah,
extra stuff.
right. Nice.
What if you CRISPR'd all your transposons that we're aware of, if they're like a common code, then you would just stop mutating, aside from random error, spontaneous?
Yeah, it would get rid of one cause of mutation, I guess, because yeah, there's still spontaneous mutations. You could still be exposed to things in the environment that might cause it too. But that might be a fun experiment, just to see what would happen if you had no more transposons. Probably not on people, but I don't know, goat or something. Not on people. Yeah. All right. So any questions on that? Just one example of transposons, and the effect of transposons in people. Do you know what the code says? Here's another example of transposons. Another example that you find actually in bacteria. Right. So this is actually a cool example because not only does it show an example of transposons, but it shows an example of a weird thing that transposons sometimes do. Here's a bacterial cell. And drawn within that bacterial cell are two different types of DNA. If I told you that one of those was a plasmid, which one would you guess is the plasmid? The one on the right, the one on the left?
Right.
Yeah, plasmids tend to be smaller than the chromosomal DNA. So what you find in the chromosomal DNA, at least the way that it's drawn here, that red thing represents the transposon. And right next to that transposon happens to be a gene that allows the bacteria to be resistant to certain types of antibiotics. Sometimes when transposons, Eric,
I thought advantageous genes is only in plasmid DNA, and chromosomal just are necessary for the surviving in its natural environment.
in most cases, but there are some things that might cause plasmid DNA segments to become part of chromosomal DNA segments.
Transformations.
Right.
Okay.
Yeah. So yeah, you probably wouldn't find that. There's some genetic phenomenon that caused that to happen. Yeah.
But not in like the wild type or prototypes or anything?
Yeah, like if you were to go back, if you could find like the wild type chromosome for this bacterial cell, then you wouldn't expect to find any antibiotic resistant genes on there. At least based on what we know, chromosomal DNA normally does. Yeah. All right. So transposons, by definition, they can jump out of a piece of DNA and then move into another part of it. Sometimes what they do is that when they jump out of a piece of DNA, they actually land in the middle of a completely different DNA molecule. Not only that, but sometimes when they leap out of a piece of DNA, sometimes it's not a clean jump. Sometimes they take some of the surrounding DNA with it. So that's in this example I'm going to show you here, that's what's going to happen. So sometimes transposons, when they leave a piece of DNA, they take some of the surrounding DNA with it. In this case, that surrounding DNA has a gene for antibiotic resistance. Not only that, but it's possible for transposons to jump out of one piece of DNA and to insert itself into a different piece of DNA. I don't like that. The reason why we might care about that is because what are some examples of things that plasmid DNA can do that chromosomal DNA typically does not do?
Share.
Share, yeah. So the reason why we might care about this is because once that antibiotic resistance gene is on a piece of plasmid DNA, that makes it a lot easier for this bacterial cell to share that with other bacterial cells, which means that bacterial or antibiotic resistance could then spread, which would be bad news for us for whatever antibiotic we're using. So the take-home point is this: it turns out that some antibiotic resistance genes are associated with transposons. In other words, they're just located really close to them on the DNA. And that means that that resistance gene can jump from the from chromosomal DNA to plasmid DNA. And again, the reason why we might care about that, the reason why that's significant, is because plasmid DNA is relatively easy to transfer to other bacterial cells, at least compared to chromosomal DNA.
How does the transfer process work? So let's say this cell splits into two daughter cells. How does it also take the plasmid DNA to both cells?
So the plasmid DNA, it's kind of a dance. The plasmid DNA replicates kind of on its own. It's not necessarily, at least not completely coordinated with what the bacterial cell does. Yeah. So the exact mechanism wouldn't be able to explain it to you, but it's kind of an independent thing. Yeah. Why? What were you wondering about that?
I was just like, well, what's the point if now it's not in the chromosomal DNA and it has two daughter cells and only one of them gets the plasmid DNA? Like, yeah, I can pass it on, it's a bit different, but like, it's not as good right now.
Yeah.
But they can take it with it.
You can take it with it. Yeah.
So it seems rather concerning that it'd be pretty easy to just make like a pool full of swarming bacteria, and then eventually super bacteria would come out of it. You just keep dumping random antibiotics into the pool. Is it really just that easy? And why haven't we all died from a supervillain? Or something along those lines.
Because we're pretty amazing.
Oh.
Yeah, our immune system is pretty robust. Yeah, but it is pretty easy, like in the lab, to just keep on culturing a bacterial species and cause a certain mutation to occur, as long as you put the right selection and kind of pressure on it.
So, for the transposons on a chromosomal DNA, were they always there, or could they also be left out of plasmid DNA?
Yeah. So are you wondering if this can go the other way? Like,
Yeah.
Yeah, that can happen too. Like during transformation or conjugation, that can occur. Yeah. That's probably, I think that's what these guys were asking. That's probably how that ended up in there in the first place, is that there was a plasmid DNA that helped facilitate the insertion of that into the chromosome.
That'd be homologous recombination.
Nice, via homologous recombination. Cool. Good questions. Anything else on this?
There's no way for that to cross species, right?
There is, actually. So it doesn't happen very often, but it is possible, like, for, I don't know, DNA released by one bacterial species to be picked up by other bacterial species. For the longest time, people didn't think that happened, but there are instances of that happening.
So just transformation.
Or are you thinking like from bacteria to like a horse? Like way different.
Why not? Like, what if a horse just starts glowing one day and you got plasmids?
Yeah, I don't know if there's any instances of that, like from bacteria to a completely different kingdom. But yeah, from one bacterial species to another, that's possible.
Are transposons only in bacteria?
No, transposons are in any cell you can think of.
Oh.
But since we're micro, we're just focusing on bacteria. Anything else on this? All right, let's give this a try. So this is the seminar that we have lined up for today. Looks like this. How about, so that we have enough time at the end to do the exam debrief, let's aim to get through maybe at least question number 17-ish. And then a heads up that we're going to change groups. We're going to shuffle the groups starting next time. If I don't get to you and you have a question, report it by going here. It's 6:55 or 7-ish, 17.